The following is a conversation with Dr Saman Maleki, Associate Professor at Western University with a special focus on oncology, immuno-oncology, microbiology, and essentially everything to do with the immune system and the microbiome. Dr Maleki is a pioneer and a world leader in the field of sensitizing tumours to immunotherapy through modulating the microbiome. Over the course of the hour, we talked about basically the state of the art associated with the microbiome and cancer. So if you're interested in how the cancer impacts on the microbiome and how the microbiome impacts on cancer, you should listen to this episode. We delved into what the current latest evidence is for the use of FMT in making a particular type of cancer therapy called immunotherapy better at treating particular cancers. We talked about Dr Maleki's journey from doing the first trials in this area to where they are now, which is raising funds to do much larger trials that should definitively prove that changing the microbiome can massively improve response rates to cancer therapies. We also talked about what the future might look like, and I have to say that even as someone who's got their finger on the pulse of this field, I left the conversation really enthusiastic and excited about the potential in the future. So if you've got an interest in cancer, it probably touches all of us at some point in time, you should really listen to this episode. This is Inside Matters. My name is Dr. James McElroy. I hope you enjoy it. Saman, awesome. Well, welcome to the podcast. We had really high engagement from the listeners and all the socials, so this one has been much anticipated. Maybe you could start off with a pleasure. We love having world experts on the podcast, so it really is a pleasure and an honor. So maybe you could start with, and I don't actually think I know this myself, how you got interested in the microbiome.
Yeah, that's a great question. So it's interesting. So I did my undergrad and my master's training in microbiology, and it was interesting because as I was doing my master's training in particular, I was becoming more and more fascinated by the immune system, and I also had this really big interest in cancer from the get-go. Although I was training in a very different field, but I was always interested to get into cancer and understanding cancer, and for some personal reasons. And then what happened was, I in all honesty, I couldn't just wait to get away from microbes. I was like I thought they were like boring, microbiology, is old science, if you will. Back in the day I was getting trained. And then there was not much talks about microbiome back then. So I started training in immunology for my PhD, and specifically in tumor immunology, and I did also train in cancer biology. So after my PhD, I was doing a postdoc, and it was interesting. I was working on a complete independent project from, if you think about microbiome or anything like that. And that was about biomarkers of response to immunotherapy. And these are the early days of checkpoint inhibitors in cancer that we are now giving them to patients, mostly anti-CTLA-4, some anti-PD-1 that we are giving patients. And then I was working on this project and trying to find what are the potential biomarkers? What is it that can tell us somebody would respond to immunotherapy and somebody wouldn't? And what caught my interest was that there were some early studies, some preclinical studies, and some early observation studies, mostly with anti-CTLA-4 back then, in melanoma mainly. And there was some hints that the gut microbiome might be an indicator of who would be a responder and who wouldn't be. And this is like around 2016 time. So, and I, as my background in microbiology, I got a little more interested into this and started to make sense to me because, I was trained microbiologist and switched to immunology, and the immune system and the microbes, they always talk, especially in the gut. So I started to look into this more deeply, and in spring of 2016, I realized there is a unique opportunity here in oncology, and that is the fact that microbiome is something we can change in somebody's body. Right. And we have been doing that for years and years. Obviously, different methods people have used, Some people try diet, exercise can affect you. Like obviously we have fecal transplants that have been used in clinic for C-Diff. So what I did, because I was familiar with the microbiology program here at Western, I started to talk to these guys and ask around to see what are potential opportunities that we could use something to change the microbiome in cancer patients. And then they put me in touch with Mike Silverman, who's the chair chief of infectious disease, who was doing fecal transplant for C-Diff patients that they were doing it through enema. And so I went and to meet him, and I talked to him about this potential opportunity in oncology. And Mike's great; he's a great guy, and he was like, nobody ever knocks on my door asking about fecal transplants. If anything, people just want to get away.
This is in 2016, right?
It's in 2016.
This is back when
so
just starting to build some momentum.
Yeah. So and then what happened was I was like, okay, this is something that we can do here in London, Ontario, because there is an established fecal transplant program for C-Diff patients, although it was done mostly through enema. And then what happened was the hardest part was actually go convince the oncology people, So after talking to Mike and other people in microbiology, so I went and started talking to people in oncology, which was like a pretty big dead end. Nobody would even remotely consider this. I've had many oncologists, great people, great friends of mine today. They would just walk away mid-conversation when I would tell them that I wanted to use something like a fecal transplant in cancer patients. They would literally just walk away.
Wow.
Wow.
So,
and everybody thought that I was a bit of a lunatic trying to do a fecal transplant in cancer patients. So anyway, so to me that was a great opportunity because the technique or the treatment has been done in humans before. We've been doing fecal transplants for years in humans, and then obviously cancer patients is a different beast. But at the same time, there's these patients are getting immunotherapy. Cancer patients, as opposed to what the general belief is, particularly in patients who are getting immunotherapy, like melanoma patients, these are not necessarily immune suppressed patients. So a lot of people associate the word cancer and immunosuppression together, and they say, oh, you cannot give them anything. So, but that's not true, scientifically speaking. So, anyways, it was a very difficult, I would say, year or two to get anybody in oncology interested. So what happened was I couldn't get any funding for this. Nobody would give me any grant money for this. It was interesting.
So your idea, so your idea was, can we do fecal transplants in patients with cancer?
Yeah, that they are getting immunotherapy.
What was the thinking?
So
immunotherapy.
They're getting. I was thinking because those days we were, and this is just goes back to years that we only had access to anti-PD-1 alone. We didn't even have ipi/nivo for melanoma. We were just giving them mostly anti-PD-1, and if they would actually not respond, they would in Canada would be eligible to get anti-CTLA-4, which is ipilimumab. But my idea was that can we give them a fecal transplant, change their microbiome with the goal that I would boost the immune response, the anti-tumor immune response in a very general way, and then give the patients immunotherapy and see if we can improve upon what immunotherapy does in melanoma, which with single agents about 30-40% response rate. So which is still great, but compared to what it was before, but not very great. So that was the beginning of it.
So for the listener, what you're talking about is using the immune system to kind of improve its effectiveness against cancer cells.
Yeah.
And maybe you could just give a very quick overview of immunotherapy, the PD-1, what's new, and it'd be great to kind of go back in time and look inside of your mind. How did you put two and two together to go, well, this fecal transplant might improve the effectiveness? Because you've gone on to demonstrate now that it does. So it'd be good to hear how you had that bright idea.
That's great. Yeah. So the way these drugs work, we call them checkpoint inhibitors, and they're obviously very widely used everywhere in the world right now. So immune cells, particularly T cells. These are the cells that they can find cancer cells in the body, and same also as cells that are infected with viruses, and they can single out these cells and kill them. Okay, so you need this anti-tumor immune response led by T cells it can find these cancer cells and kills them. Now, immunotherapy, what it does is that it comes and removes the brakes from these T cells, because a lot of times in cancer and other conditions, what happens is that the tumor can put in some brakes on these T cells. As these T cells get to the tumor, there is this mechanism that these T cells are expressing a molecule called PD-1, or programmed death 1. And then what happens is that gets engaged with its ligand that normally is expressed on the tumor cells. It's called PDL1, or expressed on macrophages or other myeloid cells in the tumor. And then what it does, it really shuts down the T cells' ability to kill these cancer cells. Now, the brilliant idea that back then Jimmy Allison and Tasuko Hanjo and those people came up with was that if we block this interaction between the checkpoints and their ligands, we allow the T cells to just move ahead with their function. And that actually stand to be true in many cancers. Now we have approvals for those checkpoint inhibitors. So, but what this really is centered around T cells, okay? And as a T cell immunologist, as somebody who was trained in T cell immunology, I knew there are ways that we can potentially impact a T cell function, and an ability to be more vigorous in its action. And actually microbiome, because as I said, the highest number of T cells in the body actually reside in the gut. And what happens is that they're always interacting in this microbial community. Okay, they're cross-talking all the time. They're metabolites that is, crossing the barrier, the gut barrier, and it's affecting the T cell function. And we've known this for years and years, that the bacteria can suppress T cells, they can activate T cells. So, and the idea was that cancer, like any other disease, the idea in my head was that they potentially have, and we hear this all the time, we're saying that cancer patients have a dysbiotic microbiota, so the microbiome is not healthy. If you think it's a chronic condition, it's like any other chronic condition, that the microbiome is really diseased. And we see that actually in our patients across different cancers too. So the idea was that can we get that diseased microbiota and replace it with something that's healthy, okay?
Yeah.
And if that change can jumpstart the T cell function, and now you layer a checkpoint inhibitor on top of that, you're really giving that T cell what it needs to go and fight the cancer cells. That was the general idea, which I think we luckily and thankfully we showed to be true, in at least in melanoma, and we're now doing it in other cancers. So what the goal was to be able to do this also in humans, because by then there were some mouse studies, there were some observational studies that are coming out, and they're like building on this momentum of there might be a link between microbiota and cancer. But I'm always interested as a pragmatic scientist, as somebody who's translational. I want to see you can do this in patients, and we can actually do it as an intervention, as a treatment. And the easiest way we could do this and show it was through a fecal transplant. And now, back then, my idea was I use somebody healthy, and we had that resource here. You go to a healthy donor and show we can do a transplant. Now, nobody would obviously give me funding for these things back then. So I got a call from a friend of mine telling me that there's a foundation that they had a call for some treatment, some alternative medicine treatment for cancer, and they want to call them. And I did. And then they got interested in the idea, and we wrote a grant, and we got our first batch of funding that got me to do like 4 or 5 patients. And then all the patients became responders, they did great with the response. Okay. And then the news was out now. And people were calling me, and then we were able to go team up with other researchers and write big grants. But I can tell you that those early days, even some of the some of the household names in microbiome research today, that they have been doing microbiome research for years and years. I talked to them, like in 2016, 2017, they did not believe you can do an intervention with microbiome in cancer. I've had people
Wow.
I don't name names, but they're like household
Sure.
names in microbiome and oncology, and you talk to
Wow.
them today, they're all behind this, But and
Yeah,
I
of
went
course.
to them
They
and
want
I
to
talked to
collaborate.
them, and they're like, oh, you're out of your mind, because like the checkpoint
Wow.
inhibitors is working 30-40% response. What if it doesn't work and makes it worse? And I was like, well, we shall see, because
That's
I'm going to do
interesting.
it. And they thought it was a crazy idea. But
I love that.
it's interesting because there was also the two other groups that at the same time, maybe actually they started a little later, but they had the resources to probably finish their cohorts a little earlier than us, although they didn't finish all the accruals because of COVID. They went and published, but there was a team in Israel that they did fecal transplant from previously responder patients into melanoma patients who were refractory, and there was a team in Pittsburgh, they're all good friends of mine, and they used also responder patients into refractory melanoma patients, and that they published their papers in Science a couple of years ago, and they showed that it was they could get about 30% sensitization, which is great proof to me that this can also work in a very difficult refractory setting. So we went obviously to the first line setting, because back then I was also thinking that microbiome, you want to go with this thing before the immune system is completely burnt out. So through different lines of treatment. And I still believe that if we actually, and I'm pushing this now more and more to the neoadjuvant setting. We just got funded from CIHR to do a trial in neoadjuvant setting breast cancer, triple negative patients. So we got some bridge funding, and we went back to get more funding. And we're designing that study that we can do this FMT in the neoadjuvant setting before the patients start to get pembrolizumab, which is immunotherapy plus chemotherapy, and see how much we are actually improving pathological response to treatment. I'm a big believer that with these type of interventions, you want to go earlier before, like you potentially get some effect later on as well, but you want to go earlier where the immune system is not completely burnt through all these different lines of treatment.
So I think what you're saying is that the other groups were giving FMT once the immunotherapy had already failed.
Yeah.
And that's in 60%, 70% of patients it doesn't work?
So
is that correct?
Yeah, in the single agent anti-PD-1, it's about 60%. Obviously, today in melanoma, the practice is different. We are giving patients ipi-nivo, which we know works. There is a much higher response rate, it's about 55, 56%. And 5-year survival, you have about 50% of the patients or more are alive, which is great, and we're doing trials.
That's great.
We have done now one phase 2 trial, which we had 20 patients in one of the cohorts in that trial. Patients received ipilimumab and nivolumab and then FMT beforehand. And we presented that data at the AACR, showing that we had 75% response rate in that cohort that we gave FMT to the patient's first line setting. And now we got funded to do a randomized trial. We're going to do a randomized trial in Canada with CCTG, which is the Canadian Cancer Trials Group. And we're going to give patients FMT plus immunotherapy or versus immunotherapy alone in melanoma. And it's going to be probably 16 sites across the nation. And we do some comprehensive translational studies to show in a randomized setting that this is going to be effective in melanoma.
Because I guess some people are still, based on the people I speak to, the interactions I have, they're still on the fence about the microbiome being important in cancer, and some people just more generally. What level of evidence, what do you think it's going to take for there to be a total transformation within the field that you're in and more generally?
That's a great question. And it is fair. It is fair because I still believe that we are in very early days of microbiome research in oncology per se, obviously microbiome research in general, because as you know, in the only last decade or so, we have had now access to all this sequencing technology and learning about all the microbes that we can't even culture, that they are there. And like we obviously did the metabolomics, and now with these new imaging approaches, that you can do FISH 16S, and you can actually see the bacteria is present within the tumor, for example. So what it really takes for in an oncology setting, besides doing all these pioneering and exciting phase 1 trials that our group and some other groups are doing, is to do a randomized trial. Like anything else in oncology. When you do a randomized trial, which we are doing here, and there is one that's happening also in Europe, I believe. I think it's called the Picasso trial, but they're doing FMT via enema. So we are doing FMT by the way via capsules, or capsules, as you're aware. So these are what's going to provide a level of evidence that people need to see, let's say, in oncology. But I got to tell you that the times have changed, and it takes time to educate everybody. Obviously you have your first group of people in the academic centers, and then you go to the community, and try to train people in the community. It's like the same thing that happened with immunotherapy, but it came out. Not everybody was, believing that immunotherapy is going to be effective, but it became a dominant force in oncology. So
right.
And I think that what's happening with microbiome is that as we are generating more clinical evidence and we're getting better at science, people are going to see more application and utility in oncology per se, but I am focused, and I'm sure in other diseases as well, as you guys are doing and other people are working on. But the key here is that for us to generate, first, the quality level data, and that's through clinical trials and randomized trials, and that's what we are trying to do, which is not easy, but you have to do it. You have to do it and show that this is really going to be impactful for oncology patients. And remember, with the fecal transplant, we can talk about toxicities, but generally speaking, at least in the context of single agent anti-PD-1, it is a pretty safe treatment, and that's not something you have common in oncology. A lot of oncology drugs come with this,
absolutely,
barrage of toxicity that they're bringing you. Now, I gotta say, we have done now 2 trials with Ipi/Nivo and FMT, and you get some other toxicities like some colitis that's going up, but generally speaking, still manageable within the same range of what we see with those combination treatments. But at the same time, we are seeing much increased response rates in patients. So the other thing that's very important is that in oncology, like when people talk about cancer, they think like this is just one giant disease, but it's not like that. When you talk about cancer, there are hundreds of different diseases, even within the same subtype. For example, when you talk about breast cancer, a patient who's diagnosed with stage 1 breast cancer is very different than a patient who's metastatic breast cancer and effectively not responding to any line of treatment now. So, what happens that these are all very nuanced, the study that you're designing, the treatment plan that you have, and that's, I would say, fertile ground for training and research. And the beauty of what I like about microbiome research that you do by nature need interdisciplinary teams. You need different expertise. You need immunologists. You need microbiologists. You need microbiome experts.
100%.
You need oncologists. You need dietitians. You need everybody to come to the table, and that has been a bit of a hindrance for a lot of teams. But luckily, our team, because we got into this early, when people became interested in our center and other centers in Canada and internationally, now we have built those teams around us that we can do these things.
Absolutely. I mean, you've certainly established a very significant reputation, I think, as one of the leading players in the space. Maybe you could talk a little bit about the journey that you've been on since the first study, and what you looked for in the first study, what you learned, and now what you're applying all your learnings to looking forward. You talked about the randomized trial now to increase the level of evidence. How did it start after that first trial? And you got published, obviously, and now you're doing more work. So you just talk the listener through that journey and what's coming next.
It was an incredible journey in sense that it was a very educational experience for me and very humbling experience, because a lot of times as scientists, for example, fundamental scientists, we think we study cancer, we understand cancer, and that's not true. And cancer is this, like this disease that has clinical aspect to it. It has scientific aspect to it. There is emotional aspect to it. So, and then the other thing that I learned is that to be a true translational scientist, it takes more than just, saying that I'm doing stuff in my lab and I'm maybe getting some patient samples, and I'm helping some clinicians. No, you actually have to lead the way, and you have to, it's very difficult because you have to become the bridge between the clinic and the science. So what the way it worked for me, and it's understandable, not a lot of people, the word translational research is a big word everybody uses, but almost nobody really practices. So I can tell you that I spent countless amount of times in the clinic myself. Today, people in my lab are doing the same thing. Even my postdocs, they're going to the clinic, they're spending time with the clinicians, or I have clinical fellows who spend time in the lab. You have to go, you have to learn what's happening when you're like trying to be part of this team that you want to design a study that's going to have multiple components to it. Like for every aspect of that first trial, and we learn more and more as we progress to different cancers, and with other trials. But to establish, for example, how you want to do the bowel prep before FMT, how much engraftment time you want to give, like I remember this was a lively discussion. We set up a meeting. We went to St. Jude's Hospital with our oncology team to meet with the microbiome team, and then like there was comment like from Mike Silverman saying that like you need to wait for engraftment, otherwise why would you even do the FMT? But on the oncology side, John Lenahan and others would say, we can't wait, we can't delay the cancer treatment for a patient with stage 4 melanoma. This is, their life depends on this.
That's interesting.
So, and it was the back and forth, and I had to put my foot down and say, we'll do 7 days and we'll wait for 7 days. And then after 7 days of FMT, we'll do immunotherapy. And believe it or not, anybody who's building trials, that's the kind of standard now. But that's how I came up with that number.
You mean FMT first?
FMT
That's
first,
interesting.
wait 7 days and do immunotherapy. But that's how, like people who don't know how the 7 days became standard for these trials, that's the story behind it. Because
there you go.
The microbiome team wanted like 2 weeks, 14 days or longer. The oncology team wouldn't want to wait. I had to pick some middle ground, and I said 7 days it is. And then we went with that.
So could we just have a little debate about that just for fun? So like, would the immune system stimulating effects of the FMT not start pretty much as soon as it hits the body, independent of engraftment?
Yeah. That's true. It can happen. We have actually done samples in patients that we got right away. Like we got the samples we got was 7 days actually post FMT, and we saw good immunostimulation peripherally. In the peripheral blood, we saw a lot of immune activation. Mm-hmm. Having said that, the best indication we've had so far, which became surprisingly a very strong biomarker, has been the success of engraftment. So engraftment in our trials have been able to predict responders with 100% accuracy. So not even 99%.
Wow.
So that's why there is more to it. And it's not just that initial, I'm just giving...
That's interesting.
Because what happens is that the idea is you're giving a lot of billions of bugs. They're coming in. Obviously, the immune system gets stimulated. But that doesn't necessarily translate into... If these bugs have successfully engrafted, producing metabolites that are going to now kick in the right anti-tumor immune response that you need for a checkpoint success, So you do need at that time for success. And the best preclinical data that we've had was 2 weeks. Frankly, it's 2 weeks, and we do 2 weeks in our preclinical experiments. We actually, before we do FMT in mice from either mice or patient samples, what we do is that we treat them with antibiotics, and then we give them FMT after like 3-4 days after the end of antibiotics for a washout period, and then we wait 2 weeks before we inject them with the tumors or get them immunotherapy. So because we want to make sure that engraftment and the microbiome has this ability to start kicking. I was in a conference, I was giving a talk, and somebody asked me, it's like the preclinical data showing 2 weeks. is the best time. Why didn't you do that in patients? I was like, I'm treating patients. You're treating patients is mice. So you have to understand the constraints of doing this in the clinic. Not everything's going to be clear cut in the clinic when you want to translate these into patients.
You've started to dive into two things that I've got written down to discuss. One is how good are animal models in oncology and microbiome research based on your experience? And then the other one is, and this is probably a tricky one, is how does FMT work in, making all of this happen? That's more of a funner type question because I know it's still a moving target, but how good are the animal models? And you've done work on classical animal models, but you're also doing work on what you call avatar animal models as well, right?
So animal
it'd be great to hear about both.
Are models, and every model is wrong. That's just something to I want to start with in anything, like when the people were doing models for COVID, the good scientists would come out and say that all the models are wrong. And that's true here as well. The models that they're showing us, but they're good tools to give us bits and pieces of objective knowledge that like something's happening, something that we can change and something that we can see. I can tell you that one of the problems, generally speaking, for example, when we do human to mouse FMT is that you're not going to have a complete successful engraftment of all the human microbiome into animals. As we've done this in our lab, we've looked at the microbiome changes. You do shift the animal's microbiome. You do actually make it closer to the donor, whatever donor is. It's a healthy person or a patient, but it doesn't completely recapitulate that donor's microbiome. Having said that, in the context of immunotherapy with tumor models, we do see that with some level of, fidelity, these models recreate what happened with the patient. So, for example, if you're giving them FMT from a non-responder, there is a higher likelihood that the animals who got the FMT, who got that donor's microbiome, you're giving them a tumor, and then you treat them with the same drug the patient or same type of drug the patient received treatment with, they won't respond. Those are the avatar mouse models, It's about 75 to 80% fidelity there, which is not bad actually, but it's not complete, And then the other animal models that people use, obviously, in this space is germ-free mouse models, and a lot of times those are useful if you're doing, a monostrain colonization and things like that for mechanism. I gotta say though, I'm not the biggest fan of germ-free mouse models. When I'm reviewing a paper for some big journal and all the data comes from germ-free, I actually make them to go use it do it in SPF mice because no human comes with, being germ-free.
Yeah. So, sorry for the listener. Could you just talk a little bit about the avatar, the SPF, just so they understand?
Yeah. So the avatar mouse models really is where you're getting an animal. It could be a germ-free, that means has no microbiome, or it could be SPF, that means we're keeping them in our facilities up here. They have their own mouse microbiome. Okay. So with the SPF mice, what we do is that when we want to make them avatar mouse mice, we deplete them native microbiome with antibiotics. We give them a broad spectrum cocktail of antibiotics, which doesn't completely get rid of everything, but brings down the load of the microbiome. And then we now hit them with a week of FMT that we do from, let's say, that patient's stool. And then what we do is that we're obviously collecting stool from them and seeing that how their microbiome goes from what it was to what it is, and now how close that resembles that human donor's microbiome. Okay, that creates a mouse avatar that is like an animal that has a human microbiome in it. So, and we have been using that.
And in the most recent work that you published in a really high-profile journal, you combined your human clinical study with animal models to strengthen the robustness of the data. It was really nice.
Yeah.
So maybe we can jump into your most recent work and then we can talk kind of more fun questions about how does this work and is a consortia better than FMT and these kinds of things.
Sure. So our more recent work in terms of, obviously we did work in, our first trial, the MIMIC trial, was in melanoma, stage 4 melanoma patients, advanced melanoma. We published that in Nature Medicine last year. And then right after we we opened that trial. I started to work with one of our GU medical oncologists here, Ricardo Fernandez, and we started this exciting trial in renal cell cancer or kidney cancer, where we gave patients multiple FMTs, not just one, and then they received combination of immunotherapy with ipilimumab and nivolumab, anti-CTLA-4, anti-PD-1. So, and we had 20 patients that we accrued. Actually, accrual is complete now, and we are now doing the translation studies on that trial. After that, I teamed up with Bertrand Routy from CHUM and his team, and we started this trial called FMT Luminate. So, and that was a one-arm phase 2 trial that had 3 cohorts in it. One cohort was advanced melanoma, where patients got FMT, a single dose of FMT, and then Ipi/Nivo. We had 20 patients in that cohort. That's complete now, and it was 75% response rate, as I mentioned. We presented at AACR. And we had one cohort of non-small cell lung cancer patients who got FMT plus pembrolizumab. These are the patients who have PD-L1 high tumors. That means they're qualified to get immunotherapy for their lung cancer. Very, very exciting response in that trial. Stay tuned for that. The 25 patients on that, I believe, is accrued. So far, the results is very promising. And then we also had 5 patients with uveal melanoma that we treated with FMT and immunotherapy. And that's, it's a very different disease than cutaneous melanoma, and they don't really necessarily respond to immunotherapy. And I believe 2 of those 5 patients actually responded, which is quite remarkable. It was just a small cohort. We just wanted to see if we can treat those patients safely. So after that trial,
amazing.
We worked. I worked with John Lenahan again, who also sees GI cancers, here to open a trial where we would do FMT plus chemotherapy in advanced pancreatic cancer patients. And I'm happy to announce it here for the first time that trial is now up at clinicaltrials.gov. So
awesome.
It went on today, actually, this morning. So
awesome.
The goal is,
congrats.
Thank you very much. That's it's always great to see another one of your brainchilds, come to
100%.
Now I can tell you, that was a very unique experience because of the nature of pancreatic cancer and how difficult that cancer is. It took us over a year to just design that protocol because of the complexities of that cancer and how difficult that cancer is. But the patients are going to get FMT, and then we give them gemcitabine or nab-paclitaxel, and then which is chemotherapy, and that's going to be 20 patients. And now we are also funded and working on a protocol to open that phase 2 trial in the randomized phase 2 trial in melanoma that I mentioned earlier in the podcast. We're going to do a nationwide trial in melanoma. It's called ME17. Today is, or this weekend's the CCTG meeting. They're going to discuss it. So hoping that later this year we'll open that trial, and that would give us some randomized trial level data about the importance of microbiome in oncology. So at the same time, I'm working with 2 other cancer centers, as I mentioned, to do a study in a neoadjuvant breast cancer setting where we would give patients FMT, and then we give them chemotherapy and immunotherapy and see how we are improving if they're improving the pathological response, and also ctDNA clearance, because that's a biomarker in that setting. And then I'm working with my friend Marco Ruella at the University of Pennsylvania to do a trial of FMT in patients who are receiving lymphoma patients who are receiving CAR-T. So we are going in that space. That's an exciting area, and we are in discussions to do a trial.
Just before you keep going, sorry to cut you off. Sometimes it's difficult when you're, doing it virtually. You're doing a lot more than I actually even knew about. I'd just like to take you back just quickly to the chemotherapy side of things and having FMT plus chemo, because we've been talking about immunotherapy. So maybe you could just explain what the rationale might be there, and then let's talk also about CAR-T, because that I've not heard about before. That sounds awesome.
Yeah, so the chemotherapy in pancreas cancer is, so the pancreas cancer, as you know, is a very difficult cancer. There hasn't been much progress in this cancer the last 50 years. And so there is some really good scientific evidence that the microbiome is involved. The bacteria are involved in from the initiation of this disease into also there is some good level data about tumor microbiome that is affecting the patient's survival and response to chemotherapy. So there was this paper that recently came out that showed that the bacteria that's involved in gingivitis, it's an oral bacteria that can translocate to pancreas and can work with KRAS mutation in these cells and cause or induce this cancer development. And then there was some really nice little data that was published in Cell a few years ago by Florencia McAllister, my good friend from MD Anderson, that they showed that patients who were long-term survivors of pancreatic cancer, they had a more diverse tumor microbiome in their pancreas.
Oh, interesting.
And then when they did FMT from those patients into mice, into avatar mouse models, they could control tumor growth and with their microbiome. And then they also showed that with the FMT, you could actually change about 25% of the tumor microbiome, which is very exciting if you think about it. And
how is that possible?
Yeah, so that's the mechanism of that is highly debated. But in our case, obviously, when they do in mice, they're doing oral gavage, remember? So they're doing the oral gavage. And in our case, we are doing FMT via capsules. So it's an oral way.
Yeah.
The capsules are going down. So we are hoping that we should be able to also change, in theory at least, some of the tumor microbiome with that. And then the third factor in pancreatic cancer is that there's a bacteria called gamma protobacter family that they live in the tumor of top of north of 70% of pancreatic cancer patients. And that bacteria eats gemcitabine for food. So, and gemcitabine is a common chemotherapy we use in that disease site. So, and think about it.
That's crazy, isn't it?
Yeah, you're loading the patients with a drug
crazy.
that tumor has a bacteria that is just going to degrade it. And so the idea is that if we change the tumor microbiome somewhat,
it's incredible.
And then we can effectively boost that gemcitabine response in those patients. And unfortunately, with this disease being so difficult and the survival rate is so low, the bar is so low that we're hoping that if we are not going to see too much toxicity and the safety signal is not that bad, we might be able to push the bar a little more. And as you know, pancreatic cancer is like, there's a lot of drug development drugs go to die, and no company wants to give you drug. Nobody gave me immunotherapy for it. So I can tell you, the goal is to do this with chemo, and hopefully if we get good data, then we can go knock on the company's door and ask for getting immunotherapy for that disease. But I normally just don't back away from a disease that's hard. That's not. I'm hoping that I can show that I can make a difference. In fact, this morning, I showed my son the clinicaltrials.gov page about. He's 8. He's going to be 8 in September. And he asked me, what is it that you're showing me that? I told him this is a gift from your dad to humanity if it works. It works. We don't know, but it works.
Fingers crossed.
Yeah.
Fingers crossed. Okay. So, I mean, in terms of this bacteria chewing up the chemotherapy, I mean, that's just kind of crazy, isn't it? You load the human up with it to try and basically suppress the growth and of the cancer cells. And it must have been such a revelation to understand that there were bacteria living inside of the tumor. Not only were they living inside of the tumor, they were eating the chemotherapy and not having any negative effect. In fact, must have been a positive effect. So that's, for the tumor, exactly. And it's just one of the mechanisms so some of the cancer cells have a symbiosis with some of the bacteria, and the bacteria help them. That's where all this comes about. It's incredible. So bringing us back then a little bit. So in terms of CAR T, which you mentioned there as well, that's a really exciting area. So these are like engineered T cells, right? Bringing you right back to the start. So maybe you want to tell a listener a little bit more about CAR T, why it's exciting, in the field of oncology, and how FMT might potentiate that as well.
Yeah, so that's also, as I said, it's a very exciting area, and it's mostly practiced right now as standard of care in hematological cancers. These are blood cancers like lymphomas and leukemias. So the CAR T is when you have a patient that's coming in with, let's say, ALL, acute lymphoblastic leukemia, and then they take some of the patient's blood, white blood cells, and they engineer those cells to express a CAR component, which is a chimeric antigen receptor for generally now for CD19 or BCMA, whatever target that we have approved for. And then what that does is that it makes that T cell, and the CAR component is really a pieces of antibody that's engineered on top of the T cell, and that it can make that T cell specifically recognize that CD19 molecule that's expressed on the leukemic cells or on the lymphoma cells. And then the T cell can be very potent because you now mass produce the T cells outside the patient's body, and then you do what we call lymphodepletion of the patients. You give them doses of chemotherapy to get rid of their immune system, to bring the loads down and also load the disease down a little bit, and then you infuse them with the CAR Ts. And then those T cells normally go, and as soon as they see the target, they start to proliferate even further, and they produce this massive response that has been very successful in fact, putting patients in long-term remissions, if we don't want to use the word cure in oncology anyways. So now, but there have been some good studies now in the CAR T space, and led by my very good friend Marco, and they've done observational studies that he published in Nature Medicine a couple of years ago, and showing that a lot of these patients have dysbiotic microbiota, and a lot of these patients, as you know, in this process, they are exposed heavily to antibiotics because they're more prone to it. These are actually patients who are truly immunosuppressed in some levels. And then he showed that the patients who had more dysbiotic microbiome, they actually had poorer response to CAR T therapy, and they didn't benefit as much. So clearly there is an evidence there that the microbiome can be important in the context of CAR T therapy. Obviously, we need to do an intervention to see prospectively if we can change the microbiome and make these drugs work better, these live drugs, which are T cells, work better. And this goes back to what I mentioned at the beginning of the podcast. As a T cell immunologist, I think you can directly change the T cells attributes by changing the microbiome. And this is no difference because in the context of checkpoint inhibitors, we're working with endogenous T cells, but these are synthetically engineered T cells outside the body, or modified T cells. But at the end of the day, they are T cells. So the idea is that if we can give the patients FMT before they are going for their CAR T, and then see maybe we give them another FMT afterwards, after the infusion with CAR T therapy. And we're working now with that team. We obviously have to go to the FDA and submit an IND application for that. But I think this is going to be very important for the field as well, to show that not only microbiome interventions are important in the context of endogenous immune response, but also where you go with cellular therapy, which, as you know, is expanding massively right now. And we recently had
huge, isn't it?
TIL therapy approved, which is tumor infiltrating lymphocyte engineered outside the body, and that got approved for melanoma. And I've already talked to Patrick Wu, who's the president of Moffitt, to see if we can do a trial with TIL therapy patients in Moffitt. So there's obviously microbiome. I think it's very important. There's lots of exciting opportunities for us in oncology, but we also have to learn a lot. There is a lot that we need to learn. As I said, every cancer, every disease population is different, and every treatment combination that you want to do might be different for these patients.
So you've actually opened my mind up to a whole nother world of opportunity, which is any emerging novel cell therapy or immunotherapy or whatever it ends up being, or novel form of chemotherapy or ADC, antibody drug conjugate, just for listening.
Yeah, ADC, absolutely.
Maybe the microbiome could potentiate all of these things. And if so,
you have something that has got systemic effects. Yeah.
It's incredible. So we've got about 10 minutes or so left. I wanted to now ask you about what the future holds. So you've talked about CAR T. You talked about immunotherapy. What is your vision for how all of this translates to the next step, which is bigger studies?
Yeah.
Some of the listeners might be thinking, why are the big companies not jumping all over this?
Yeah.
So, what, which we've discussed obviously many times, because it feels like we're on the precipice.
Yeah, no, that's obviously a logical question, you know.
Yeah.
So what obviously in industry, you know, it depends on the industry you're looking at. As I said, microbiome is a very novel thing, especially now in oncology. And you got to think about oncology is its own very established industry. When immunotherapy entered, or immuno-oncology entered oncology, it was this little thing which is now over maybe 70% of all oncology is immuno-oncology. So, and I believe with microbiome is the same thing, that we are going to have an explosion of interest as there is more and more science, better science, and more clinical evidence is built. You know, the way I think about things is that I'm unfortunate and a person who's not very patient, you know. So, and I go around and ask people, can you do this? And they say no. And then unfortunately, I have to go build it myself. You know, some people, in the context of industry, you know, some people are company builders, but some people are industry builders, you know. And I see myself as somebody who's helping to bring this into oncology in general, because I personally, based on the data that I see, obviously everybody has their own biases, but I believe there is tremendous opportunity for cancer patients and how we can help them with just changing their microbiome. And we talked about obviously primarily FMT today, but there is a lot other that can be done, you know, like food spectrum consortia, food spectrum probiotics that people are working on, you know, limited consortia, diet, exercise, everything that's happening, you know. And but we primarily talked about FMT obviously for obvious reasons. But I do believe there is a lot of opportunities in oncology, not just for us to improve response rate, but also maybe to reduce toxicity, to manage effectively patients' conditions that are going through all these stages of treatment. For example, in breast cancer, we know that there is a lot of mental health components into this after patients are receiving chemotherapy, and that they can develop depression. So I told my team here that when we do this FMT study, we should also look into, because there is this established gut-brain axis, you know. So see if we're changing the microbiome and patients are going through this treatment, chemo treatment, are we changing their mood? Are they feeling better going through this journey with chemotherapy? So obviously there is a lot, and this is all multidisciplinary, you know. The problem is at the end of the day, I'm one person, you know. So I need a lot of people to help me in what I do. I come up with some ideas, but there's a lot of great people that come in and help. Otherwise, none of it can be done.
Thank you so much. Could we just have a quick discussion about maybe some of the future cancer indications and why some cancers are better than others? And one of the next kind of obvious questions that people might have is, well, is this going to be given to every cancer patient at some point in the future, whether it's after chemo, before chemo, pre-immuno, during immuno? And how far away are we from that being a reality?
So those are obviously great questions, and something that me and my team always are thinking about or discussing internally and externally with collaborators and partners and, you know, scientists and companies. And so I do think that there are certain cancers that are more prone to improve response. Obviously, I think melanoma is a no-brainer at this point. That's why we are doing a randomized trial. In the context of GI cancers, there are potential opportunities. For example, you know, the effect of bacterial nucleum in colorectal cancer, and can we do an FMT? And this is a cancer that primarily doesn't respond to immunotherapy and sensitize it to immunotherapy. You know, there is this opportunity that I don't know how much people know about or think about, but that is an immune response towards microbial antigens. That when you do an FMT and if some of that bacteria translocates to the tumor, particularly in GI cancers, and now you actually induce an immune response against the bacterial antigens that are not present in the tumor. So you don't have a originally activated anti-tumor response, you have an antibacterial response, antimicrobial response. but this was effectively initiated because of that FMT or the microbial therapy, especially in the GI cancers where this is going down and the cancer can be colonized directly. I see a tremendous amount of opportunity in those cancers. Another cancer that people don't think about, but we are thinking about right now, I looked at the presentation we're going to present at CCTG with one of my partners in Ottawa, is brain tumors and GBM, glioblastoma.
Wow.
And why we are thinking about glioblastoma, as we talked about, there is tremendous evidence about gut-brain axis. I reviewed a ton of papers about how metabolites, microbial-driven metabolites, make their way into the brain. And the idea with that would be, can we do FMT and then change this massive immunosuppressive tumor in the brain of these patients that they're getting? So I do think there is a lot of different cancers, and there's good scientific rationale for a lot of them that we can treat with FMT or other modes of treatment with microbiome interventions, because you said it can be before, it can be after, it can be for patients in the preoperative setting or patients in the adjuvant setting. These are patients after surgery, or patients who are just going through chemotherapy or immunotherapy, and we want to give them maintenance of their gut microbiome. The other idea is that something that we are doing in our trials is that we give patients rescue FMT after they have to get antibiotics. So that's an important area as well. So think about it. What percentage of cancer patients, as they're going through their cancer journey, are treated with antibiotics? This is a big number.
So many, right?
Yeah.
So many.
And we know for a fact that the clinical evidence is so overwhelming that the antibiotics are going to be harmful to that patient's response and survival for the treatment of the drug they're getting. That's just logic there to give the patients FMT, at least if they go antibiotics, So there is tremendous opportunities to help patients. The only thing is, obviously, in medicine, the clinical practice doesn't move as fast as science, So and that's why we have to all work together, and somebody has to come around and translate these things. And that's some of the things that I'm doing, and there are great scientists out there that are doing as well.
Saman, so much to be excited about. We could have kept going, I think, for another hour about mechanism of action, consortia versus full spectrum, single strain, so on. But we're at the hour now, and I wanted to take the opportunity to thank you for coming on, but also thank you for all the hard work that you're doing, pioneering this really, really exciting new field of science and medicine. Now, if the listeners want to follow you and follow your work, where's best to find you?
Okay, so thanks, James, for inviting me. It's always great to talk to you. They can follow me on Twitter, as my handle is, or X, SM Immunology. And they can also search my name, and I'm on LinkedIn, and I interact with people on LinkedIn. I apologize if people send me a message. I get hundreds of messages on LinkedIn. I don't get to always answer all of them, obviously. But if they search my name, we have a laboratory website, and the type of work that we are doing, it's easy to find.
Thanks so much. No, this has been awesome. I'm a believer, as you know, but I've actually finished the conversation here even more excited about the opportunity than when I started. So you've done a great job enthusing me, and definitely done a great job for the listeners. So pleasure, and keep up the great work. Thank you so much.
Thanks, James. Great talking to you today.